Every single living thing is made up of something that's called DNA. Your DNA is like an instruction manual with the directions for how to build you. Your DNA decides how tall you're going to be, your hair color, your eye color, and more. Sometimes, while the directions are being written and copied into the instruction manual, mistakes can accidentally happen. These mistakes are called mutations. Sometimes these mutations are helpful. For example, there's a mutation that changes the color of the fur of mice from brown to white. If the mouse was trying to hide on a beach so a cat wouldn't be able to find it, the white mouse would hide much easier than the brown mouse would. Unfortunately, mutations can also sometimes hurt people. They can cause many different diseases, such as cancer or something called Down syndrome. People with Down syndrome have many different symptoms. They usually won't grow as tall as people without it. They also may have heart defects, which means that their hearts don't work as well as they should, and there's a lot of problems there. Or they also might have poor hearing or learning disabilities. The learning disabilities might make it a lot harder for them to learn in school in doing simple things such as reading. Every living thing is made up of trillions of cells. Each of your cells contains DNA, which is then further condensed into something that is called a chromosome. All your chromosomes and cells are constantly making identical copies of themselves in order to build all the parts of your body. Sometimes mistakes can happen in these processes, and this is known as a mutation. Sometimes the mutation occurs in the DNA sequence itself. If mutation occurs in your DNA, the result can range from no effect at all to serious diseases such as sickle cell anemia. So how is this possible? Take this sentence, for example. The fat cat ate the rat. Now, if you take away the period at the end of the sentence and read it again, it still makes sense. Now let's look back at the original sentence, but instead of removing a period at the end, we'll remove a letter from the middle. The first word's going to remain the same, but the rest of the sentence doesn't make any sense. This would be an example of a mutation that would have a more serious impact on your body. Mutations can also occur on the chromosomal level. There are 4 different types that could occur. The first is deletion, which is when a part of a chromosome is missing. This means lots of genetic information will be left out, which could lead to many different consequences. There's also duplication, also known as insertion, which is when a part of one chromosome splits off and joins another chromosome. As a result, the chromosome will have extra copies of the same genes. Chromosomal inversion is when part of a chromosome breaks off, then reattaches backwards. And finally, translocation is when one part of a chromosome breaks off and joins onto another. The result is uneven-sized chromosomes, with some being a lot longer or shorter than others. All of these mutations can cause a wide range of conditions, from colorblindness to sickle cell anemia and to Down syndrome, which is when there's an extra chromosome in your cell, or extra chromosome on your 21st pair of chromosomes.
Mutations can occur during meiosis in chromosomes, or they can occur during translation between nucleotides. Point mutations are the errors that occur on a single nucleotide base. A single change in a nucleotide doesn't sound like much, but it can set off a chain reaction that could affect the way the entire protein functions. Nucleotides are read in groups of 3, called codons. During translation, which is facilitated by ribosomes, these codons code for a specific amino acid to be added to a growing peptide chain by tRNA. When the chain of amino acids has been completed and translation has been terminated, the protein is in its primary form. To become a functioning protein, it will begin to hydrogen bond with itself, which will result in its secondary form. Next, bonding between the R groups of the amino acids will result in the tertiary form. And finally, many tertiary proteins will come together to form a quaternary structure, such as this hemoglobin, where it'll be ready to perform its function as a protein. So why does why does a single nucleotide error matter? In some cases, it doesn't. Silent mutations occur when there's an error on the 3rd nucleotide in a codon. This mistake usually won't result in a change in amino acid, because most amino acids are coded for by multiple codons, who vary only in the last nucleotide. If errors occur in the 1st or 2nd nucleotide, it will change which amino acid is being coded for during transcription. This is called a missense mutation. Since different types of amino acids have different properties, some being positively charged, negatively charged, nonpolar, or empathetic, This will interact differently in a protein's tertiary structure. This change in tertiary structure will affect the overall shape of the protein, which could affect the function it's meant to carry out. A nonsense mutation occurs when an error results in a stop codon, which will cause early termination of the polypeptide chain. This will result in an incomplete protein and will be detrimental to the function of the protein, especially if it occurred early on in translation. If a nucleotide is deleted from a sequence, every following codon after will be shifted over a spot, which will completely alter the amino acids that are supposed to be there. The order of the amino acids will also be disrupted if a nucleotide is added to a sequence. The subsequent nucleotides will shift over a spot and affect every amino acid in the sequence. Both deletion and insertion point mutations are a type of frameshift mutation.